phosphorylated protein kinase can intermediate the phosphorylation of certain proteins that will induce physiologic effects to the cell. We expect that in order to control the output of physiologic effects by phosphorylated proteins produced by activated protein kinase A, an enzyme called phosphatase will or dephosphorylates the activated protein. No? To show you some examples of hormones that uses the effector adenyl cyclase. Okay, here is a table summarizing some of those hormones. Okay, there are hormones stimulated by adenyl cyclase producing um, cyclic adenosine monophosphate, while there are also hormones that are inhibited by the effector enzyme. Okay, another example of secondary messenger that we would like to introduce in this lecture is the messenger IP3 or inositol 1,4,5 triphosphate. If you notice in this diagram, it is produced or liberated from the molecule phosphatidyl inositol 4,5 bisphosphate or PIP2. Okay, in the presence of an enzyme phospholipase C, this molecule is cleaved into diacylglycerol and inositol triphosphate. Now, let's take a look into how inositol triphosphate actually works. In this diagram, hormone receptor interaction results in the activation of the enzyme phospholipase C, breaking down the substrate PIP2 into diacylglycerol and protein kinase C. Similar with the effects of protein kinase A, protein kinase C also produces phosphorylated or phosphoproteins that will result or that will have direct participation in the production of some physiologic responses. Oh. Alongside with uh, um, the activation of protein kinases, okay, IP3 inositol triphosphate also induces some cellular response. Okay, for this case, we appreciate that IP3 liberated from the breakdown of PIP2 will result or will actually induce the release of intracellular calcium from endoplasmic reticulum or other organella reserves. Okay, and the increase in calcium will actually lead to some physiologic response, no? Which I'll be showing in a little while how important calcium is, no? Let's take a look at some of the functions of calcium as a mediator of hormone action. It's been established that calcium serves as a mediator of hormone action as it is seen to have many effects. Number one, when calcium is not provided, okay, let's say the media in which the hormone is allowed to act is free from calcium. Okay, it's been shown that the hormone has impeded response or impeded action. Okay, some hormones can also be what do you call this? Some hormones can also be activated. Okay, if there is an influx in the calcium, okay, to the cell. Right? An example of this is glycogen metabolism in our liver by means of hormones vasopressin and beta adrenergic catecholamines. Okay, so these two hormones require a net influx or a net increase in the amount of